Dia nak start. Okay. Okay, Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera. Good evening to all. First of all, I would like to introduce myself. Okay, my name is Muhammad Nazif bin Samad. I am one of lecturer at Faculty of Health Sciences, University Selangor, UNISEL, and the campus is located at Shah Alam. So kami dari Shah Alam ni, Fakulti Sains Kesihatan dekat Shah Alam lah. It is not at the main campus. We have another campus at Bestari Jaya. That is the main campus, the bigger campus. But our faculty in the city campus, dekat je dengan Jekyll. Okay, so the topic that I will deliver today, it is about genetic linkage and mapping in eukaryotes. Okay, genetic linkage and mapping. Okay, genetic linkage, maybe this is new term for you. Okay, in the previous lecture, I believe you have learned some of fundamental element about genetic, such as meiosis, mitosis, allele, okay, dihybrid cross. Okay, so actually that concept is actually important for you to digest before you are able to understand this topic. Tu dia punya syarat dia. Maksudnya you kena ada clear idea dulu tentang perkara-perkara yang saya sebutkan tadi. Okay, about meiosis, about crossing over, about genotype, phenotype, okay, common things in genetics. But I believe maybe some of you still not doing the revision. Mungkin ada pelajar yang tak sempat nak buat revision. Although you have attended the previous lectures. Okay, so I try my best. Maybe in the middle of this topic, I try to do what we call quick revision. Kita akan buat quick revision lah. Sikit-sikit kita revise balik apa yang you dah belajar lah. Actually, it is not necessarily what you have learned in the previous lecture. Tak semestinya. Okay, such as allele, that concept, allele concept, actually you have learned in SPM. Maybe you have learned in foundation, you have learned during your diploma. Okay, so we try to refresh. Okay, so I am hoping that you will be able to grab new concept that you will be learning today. Okay, so I would like to introduce myself. Okay, so I would like to introduce cell in my body actually. Okay, I try to take cell from my skin. Okay, ambil satu sel dekat skin saya. Okay, ambil satu and then we try to investigate what is inside of the cell. Skin cell is a somatic cell. Skin cell is a type of somatic cell. So supposedly, as a normal human, so the number of chromosome must be 46. You all pun ada 46. Okay, saya pun ada 46. 46 chromosome. Okay, so I am showing you a picture of karyotype. Ni gambar ni kita panggil sebagai karyotype. Okay, so actually it is from one cell. It is from nucleus of cell. Inside of the nucleus you can see chromosome there. Okay, and then we try to rearrange the position of the chromosome. Kita cuba susun balik posisi chromosome tu. Okay, mana yang nampak lebih kurang sama jalur-jalur dia, kita letak sekali sebagai pasangan. Okay, as example here, number 1, we can see here there are some band on the chromosome, and we try to put as a pair. Kita letak pair. Okay, so from this picture, from this karyotype, okay, based on this appearance, we can see there are 23 pair of chromosome. Berdasarkan gambar ni lah, ada 23 pasang. Okay, there are 23 pairs of chromosome, or I can say 46 chromosome. Okay, I can say 46 chromosome or 23 pair of chromosome based on this picture. Okay, jangan pula ada yang kata manusia ada 46 pair of chromosome. Itu salah lah. Konsep dia dah lain lah kan. Okay. Or you say a normal human has 23 chromosome. You lupa perkataan pair pula. Okay. So it is important for everyone, memorize, a normal human has 46 chromosome or 23 pairs of chromosome based on the karyotype, normal karyotype lah. Okay. So actually there are 23 of the chromosome here. Okay, I inherited from my father. My father, Samad, give 23 copy to me. Dia bagi, bukan dia bagi kromosom dia bagi kopi kromosom. 23 copy of Samad is given to me. Okay, and then my mother, bonda saya bagi 23 juga. 23 campur 23 cukup lah 46 kromosom. Alhamdulillah cukup 46 kromosom. Okay. So same goes to you. Okay, bila kita gabung apa yang mak kita bagi, apa yang ayah kita bagi, so every our somatic cell, okay, normally it has 46 chromosome, 46 chromosome, 23 daripada mak, 23 daripada bapa. Okay. And I have 10 siblings. Saya ada 10 adik-beradik. Okay. But the question is, why me, Nazif, it is not same like Khazani. Although we are inheriting from same parent. Okay, Khazani also received 23 copy from Samad. And he also received 23 copy from Fatimah. Same goes to Hafizan, Akrami, Zanirah. But when I see them, they are not same with me in terms of appearance, in terms of phenotype. Okay, so itu persoalan yang saya cuba ungkapkan petang ni. Any answer from the class? Kenapa? Siapa boleh jawab? Kenapa lain? Kata tadi dapat 23 daripada mak, 23 daripada bapak. Okay, nak dengar sikit suara student UITM di hatiku.
Sebab ada meiosis.
Sebab ada meiosis. Okay, so it is what happened in meiosis that produce the variation? What happened in the meiosis?
Crossing over.
Okay, crossing over. Okay, so this is actually this topic actually the continuity of meiosis. Dia cerita balik pasal meiosis, kita akan sentuh balik pasal meiosis, kita akan sentuh balik mengenai crossing over. Okay. Okay, this is two guy, ada dua lelaki kat sini. First guy on our left with blonde hair, okay, rambut perang, mata biru. Rambut perang, mata biru. Another guy here with brown hair and brown eyes. So from the observation of Caucasian population, It is common, people with blue eyes, commonly they have blonde hair. Dia ada warna rambut yang perang. Kalau orang tu dia mata coklat, kebiasaannya rambut dia bukan warna perang. Biasanya rambut dia warna brown juga. Okay. So, any idea from the class why we can observe this situation? Kenapa kebanyakan orang kalau mata dia brown, dia punya rambut dia pun warna brown juga. Dan jarang-jaranglah kita jumpa from the observation of this kind of population lah. Jarang-jarang kita nampak kalau orang yang mata biru dia rambut dia brown. Any explanation from this thing, class? Ada je orang yang mata warna brown dengan rambut perang tu ada je, tapi in term of the frequency, it is commonly we can see blonde hair with blue eyes and brown hair with brown eyes. Untuk phenotype inilah, untuk dua jenis phenotype inilah. Okay, the answer is, it is related with topic today, genetic linkage. Genetic linkage. Okay. So before we learn about genetic linkage, okay, we need to check our understanding of certain concept dululah. Saya cuba buat recall balik apa yang you belajar sebelum ni. Okay, I try to take chromosome 1, pair number 1 and pair number 7. Okay. So this one, I can say these two chromosome as homolog chromosome. Okay, saya boleh panggil dia sebagai homolog chromosome. It is from this picture, daripada gambar ni lah. Gambar ni, okay, I just crop this one. Saya cuba crop chromosome pair number 1 dengan pair number 7 here, okay, and then I put in another slide here. I want to focus on these two pair of chromosome number 1 dengan chromosome number 7. Okay. My question is, what is homolog chromosome? Apa maksud homolog chromosome? Ada siapa-siapa tahu? Okay, boleh buka mikrofon. Okay, if you write on the chatting box, sorry, I am not able to open it because my computer now is fully slideshow of my PowerPoint, so saya tak nampak lagi lah Google Meet tu. Okay, what is homolog chromosome? Kita kena check dulu kita punya understanding sebelum kita sentuh mengenai genetic linkage. Okay, what is homo? Homo class. Homo means what?
Same.
Sama. Homo. Okay, log, L-O-G. L-O-G, log. Homo, log. Homo tahu dah sama. Okay, log. Okay, everybody, you can go to Google, go to image Google and you search word log. Okay. Tengok gambar apa yang keluar. Kalau you tulis LOG, you search dekat image. Saya nak minta you buka dekat image. Apa yang you nampak? Gambar apa yang keluar?
Batang pokok.
Batang pokok. Betul lah tu. Log tu batang kayu. Batang kayu. So kalau homolog maksudnya batang kayu yang nampak sama. Okay. So batang kayu ni, kromosom ni, okay, this chromosome and this chromosome, I can say they are homolog because in terms of the appearance, in terms of band of the chromosome, it is look like same. Dia nampak sama. So that's why saya boleh letak dia dekat-dekat. Okay, from the cell, when we observe in the cell, it is not like this. Dia bukanlah duduk teratur macam ni. Dia bersimpan siur dalam kita punya cell. When we take the skin cell, kita ambil skin cell kan. So when we observe in the nucleus, it is not arranged like this. Okay, means we have to do some procedure we call as karyotyping. We have to disperse the chromosome. Maybe some chromosome dia ada yang bertindih, dia ada bergelimpangan lah dalam nukleus tu. But we try to find the best cell that showing they are dispersed, kan, dia duduk jauh-jauh, and then we try to snap. Okay, we snap the image at high resolutions, then we print out, kita cetak dia, and then you have to use scissor to cut each of the chromosome. Potonglah 46 kromosom ni kena potong, and then you have to rearrange on another paper lah, kena ada gam lah, tampal, and then the chromosome that you can see they are alike, lebih kurang sama, batang kayu tu nampak jalur lebih kurang sama, so you can mesh them. Kita boleh meshkan lah, so dapatlah this pair. Okay, so we can say this is homolog chromosome. Baru boleh cakap this homolog chromosome. Between this one and this one, the relationship of these two chromosome is homolog chromosome. And then this one and this one, they are homolog chromosome. Okay, let's say I take this chromosome. Okay, saya ambil chromosome ni, saya letak kat sini kan. Okay, I take pen number 1 here. And then I take this one. Okay, and I put aside. Saya letak sebelah-sebelah. Okay, this one I put here. Okay, so my question, can I say this is homolog chromosome?
No.
No.
So we can call this is
Heterogeneous.
Heterologous.
okay,
And
lagi sikit lagi.
Non-homologous.
Yes, non-homologous chromosome. Kira tu pandai dah tu. Ingat dah tu yang belajar sebelum ni kan. Okay, so okay, saya tak balik pair dia. Okay, so this one I get from my father. This one I get from another individual. Saya dapat daripada ibu saya. Okay, this one I get from my father. This one I get from my mother. Okay, so sebab tu kita ada 2 set of chromosome, kan. Setiap orang dalam kelas ni semuanya mengandungi 2 kopi. 2 copy of DNA. 1 copy from mother, 1 copy from father. 23 dengan 23 campur dua tu kan. Okay, so can you understand homolog chromosome and non-homolog chromosome? If I re-mention this word, so you dapat idea lah. Kalau homolog chromosome tu batang log dia tu nampak lebih kurang sama. So kita boleh kelaskan, oh this is pen number 1. Pen number 1 tu dia paling panjang. Pen number 2 dia makin pendek. Pen number 3 makin pendek. Tu cara kita susun untuk kita hasilkan karyotype lah. We call the procedure of producing that picture we call as karyotyping. Prosedur dia lah, start daripada kita campur sampel tu dengan buffer, kita ambil gambar, kita print, kita gunting. Prosedur tu kita panggil sebagai karyotyping, to produce a picture. Picture tu kita panggil karyotype. Okay, so saya buat sikit je revision. Okay, we go back to this topic. Okay, genetic linkage and mapping in eukaryote. So the learning outcome, apa yang kita expect you akan dapat bila habis lecture ni bersama saya. Okay, the first is you should be able to explain the relationship between linkage and crossing over. Okay, crossing over, ada your friend tadi sebut crossing over dalam kelas saya tanya kan. So you dah dapatlah idea lah, crossing over. Oh, pernah dengar lah perkataan tu. Linkage maybe new. Okay. But linkage in bahasa, maksudnya apa? Linkage. Linkage. It is from word link.
Bercantum.
Bercantum. Lagi.
Penyambungan.
Penyambungan, perkaitan, penyambungan, percantuman, bolehlah tu kan, link kan. Okay. Okay, and another objective is you should be able to explain genetic mapping. Apa itu genetic mapping? You sepatutnya boleh terangkan lah bila dah habis belajar topik ni dengan saya petang ni. There are two things that we are going to achieve. First, you should be able to explain about linkage. You should be able to explain about crossing over, how linkage and crossing over are related. And you should be able to explain the genetic mapping in animal. Okay. Each species of organism must contain hundreds to thousands of gene. Okay, setiap organisma dia ada beratus-ratus, beribu-ribu genes lah. For human, how many genes in human class? Berapa gene yang ada dekat dalam badan you? Try to guess, tak tahu, cuba tembak. 1 juta? 3 juta? 100? 1000? For human.
20,000.
It is around 25,000. Around 25,000 lah. Which is located in 46. 46 apa?
Chromosome.
Chromosome. Okay, good. So each chromosome is likely to carry many hundred or even thousand of different genes lah. Kalau manusia kita ada 25,000, so 25,000 tu ada dekat dalam 46 kromosom lah. So the number of gene in each chromosome, they are not necessarily same. Kalau kromosom yang panjang, mungkin bilangan genenya lebih banyak lah. Okay. The transmission of gene close to one another on the same chromosome will violate Mendel's law of independent assortment. Okay, previously have you learned about Mendelian inheritance? Ada belajar ke Mendelian inheritance? Ada ke tajuk tu sebelum ni? Yang belajar dengan Puan Hati ni. Adalah. Okay. Kalau ada yang kata tak ada pun tak apa, sebab I believe you have learned before. In secondary school pun dah belajar lah. Kalau siapa yang ambil biologi ataupun ambil sains, sains pun belajar pasal Mendel. Okay. So kita nak tengok cerita sekarang, kita nak tengok apa yang Mendel kira kan, dia guna apa, pokok kacang tu. Okay. Then we follow certain rules, certain calculation. Right. But sometimes when we do the study, we try to expand the study. It show in certain cases, it doesn't follow the ratio that has been elaborated by Mendel. Ada juga yang tak sama. So cerita yang petang ni kita nak tahulah tak sama tu sebab apa. Okay. Next, it is important for us to understand some of term sebelum kita cerita lebih lanjut pasal linkage ni. Okay, first is what is genetic linkage? Okay, genetic linkage is a phenomenon that genes close together on a chromosome tend to be transmitted as a unit, which influences inheritance pattern. Okay, certain genes they are close together. As example, they are close together, although they are transmitted to the next generation. Okay. As example, what I show in the early of the slide, and I show people with blue eyes. Okay, there are certain genes that encode to make color for blue eyes. That gene, it is close together with gene that encode to make hair with blonde color. Okay, so dia macam tak nak berpisah lah. Ada yang berpisah. Ada yang berpisah tu maksudnya that's why we can see people with blue eyes with brown hair. Maksudnya berpisah lah tu. Okay, so but most cases we can see people with blue eyes, they have blonde hair. Okay, maksudnya gene tu dia macam dia cenderung untuk diwariskan bersama-sama. Dia macam tak nak berpisah lah. Okay, so that is genetic linkages. That phenomenon, phenomena that genes close together on a chromosome tend to be transmitted as a unit. Dan dia boleh influence lah kita punya inheritance pattern. Okay, that is the first word. Second is we should know what is synteny. Synteny maksud dia apa? Synteny is a term to describe about conservation of block of order within two set of chromosome that are being compared with each other. Conservation maksudnya dia terpelihara, dia punya duduk sekali tu, dia conserved, although it is transmitted in some generations. Okay, so two or more genes are located on the same chromosome are physically linked. Okay, dia gene tu yang selalu yang tak nak berpisah tu, okay, in term of location on the chromosome, they are located close. Dari segi dia punya physical kedudukan dia, biasanya kalau yang macam tak nak berpisah ni, dia duduk dekat-dekat. They are physically linked. Okay, so that is synteny. Okay, another term is crossing over. Okay, you have learned before crossing over. Crossing over is the exchange of genetic material between non-sister chromatic. Oh, apa benda pula non-sister chromatic? Okay, crossing over is the exchange of genetic material between non-sister chromatic of homolog chromosome during meiosis, which results in new allelic combination in the daughter cell. Okay. Okay, so kita buat revision sikit. Revision sikit kan. Okay, this is chromosome, copy from my mother. Okay, this is copy from my father. Okay, buat dua-dua merah pula lah ni. Okay, tak apa. So yang ni daripada mother saya, ni daripada ayah saya lah. Okay, so these two copy I get from different sources. Dua source yang berbeza lah. Okay, so this is homolog chromosome lah. Homolog chromosome. Okay, so chromosome from my mother ni, dia apa yang berlaku dekat dalam chromosome, what happened inside of this chromosome when cell develop, bila cell tu dia berkembang, okay, something happen inside of the chromosome. Okay, apa yang berlaku dalam tu? DNA replication. DNA is replicated, DNA itu digandakan lah. DNA digandakan. So in term of composition, genetic composition dalam ni, lama-lama dia semakin banyak lah sebab dia berganda kan. So berganda asalnya macam ni. Nampak ke tangan saya? Asalnya macam ni. Okay, kromosom yang copy daripada mak saya. Lama-lama dia semakin tebal. Dia semakin tebal jadi macam ni. Okay, jadi macam double ni. Sebab dia berlaku, dalam tu berlaku DNA replication. Sel dia berkembang kan. And same goes to my father, copy from my father. Okay, satu kromosom ni, dia berlaku apa? DNA replication, sel tu dia berkembang, so dia jadi makin lama makin it is visible under microscope, kita boleh nampak. Asalnya tak nampak guna mikroskop, tapi lama-lama bila kita perhati, kita tengok dia makin lama dia makin berkembang, dia jadi tebal macam ni. Ya, sebab berlaku DNA replication. So dia makin tebal dan kita boleh nampak nak terbelah dah ni dua ni. So that's why you can see another structure like this. Kan kadang-kadang kita nampak kromosom tu dalam keadaan macam ni. Okay. Ada dia bercantum di satu tempat kan, centromere kan. Ini pun sama juga, sebab dia makin lama makin asalnya dia macam ni, tapi lama-lama berkembang lah sebab berlaku DNA replication. So when DNA replication happen, okay, it will produce another copy of DNA. Sama ke tak dengan yang asal? Yang bila dia buat satu lagi kopi tu, it is same or not?
Same.
Same.
Same lah. So I can say composition of this part, bahagian ni, and composition of this part, they are same. Dia akan sama, content dia sama. Selama-lama dia akan separate nanti. Bila dia nak pergi, when they are moving to two different pole kan, bila berlaku division, okay, it will be moving towards two different pole. So content dia, dua sel yang terbentuk tu dia akan sama lah kan, sebab it happen, what happen inside here is DNA replication. Okay, so this is sister chromatic. This is sister chromatic. This is also sister chromatic. Okay. The connection between this sister chromatic and this, this one and this one, kromosom yang ini dengan kromosom yang ni, kita panggil sebagai sister chromatic. Okay. And then this one and this one, connection dia juga kita panggil sebagai sister chromatic. Connection dua ni, connection yang ini dengan yang ini sister chromatic. Connection yang ini dengan yang ini juga sister chromatic lah. Kalau saya cakap connection between this one and this one, yang tu kita panggil sebagai non-sister chromatic. So crossing over actually exchange genetic material that happen between non-sister chromatic. Maksudnya kopi yang daripada bapa, dia exchange dengan kopi daripada mother. Kalau kita kata exchange tu berlaku di antara this one and this one, kalau kita cakap crossing over tu berlaku antara this one and this one, so salah lah. Tu bukan crossing over lah. Crossing over tu adalah istilah untuk kita beritahu exchange, pertukaran yang berlaku antara DNA daripada dua sumber, daripada bapa, kopi daripada bapa dengan kopi daripada mother. Okay, boleh kelas setakat ni? Sebelum kita pergi lebih jauh, ni revision sometimes I will make a revision. Okay, chromosomes are called linkage group. Kromosom ni kita juga panggil sebagai linkage groups. Chromosome contain a group of genes that are linked together. We have one chromosome kan, so there are many genes. So kalau so dia adalah kromosom tu dipanggil sebagai linkage group lah, sebab gene tu kan dia connected, dalam satu kromosom dia connected. Okay, I have 3 pictures here, ada 3 gambar kat sini. Okay, picture A, it show gene 1 and gene 2. Dia duduk dekat-dekat tu kan. Okay, so can I say gene 1 and gene 2 are linked? Ni gene ni dia link ke tak link?
Link.
Link. Okay, link genes mean sit together on a chromosome, making them likely to be inherited together. Kalau dia duduk dekat-dekat, kemungkinan untuk dia diwariskan bersama-sama adalah lebih tinggi. Okay, if the location of the gene are close, they have higher chance to be inherited as a unit. Okay, yang B. This one is link or not link?
Not link.
No.
Okay, not link lah. Okay, genes on separate chromosome are never link. Dia tidak link lah. Okay, C. Macam mana? Link ke not link?
Not link.
Not link.
Not link. Oh, hebat. You all dah dapat ke slide? Okay, not all gene on a chromosome are linked. Kenapa tak link? Dia kan duduk dalam satu kromosom.
Jauh.
Sebab jauh. It is considered as not linked sebab jauh. Okay, not all genes on a chromosome are linked. Genes that are farther away from each other are more likely to be separated during a process called homologous recombination. Okay, so this is another important key you learn. If the location of gene is farther away, okay, so making them likely not to be inherited together. Ada dua keadaan lah sekarang ni kan. Okay, sama ada dia nak diwariskan bersama-sama ataupun tak diwariskan bersama-sama. It depending on the location of gene. Okay, so the number of linkage groups is the number of types of chromosome of the species. Okay, for example, human, we have 22 autosomal linkages group. Autosomal yang mana? Autosomal it is beside X and Y lah, selain daripada X and Y. Beside sex chromosome, means chromosome number 1, okay, pair number 1, pair number 2, sampai pair number 22. This is one group, one linkage group. Homolog chromosome dengan homolog chromosome tu dia panggil one group lah. This is another group. Okay, 1, 2, 3, 4, 5 sampai kita ada 22. We have this one. Kalau X dengan Y ni dia tak boleh jadi group. Okay, walaupun dalam karyotyping punya karyotype we put it as a pair. Dalam gambar kita letak as a pair, but actually there are separated group. Ni group lain, ini group lain. We call as different linkage group. So we have 22 autosomal linkage group. We have one X chromosome linkage group. We have one Y chromosome linkage group. Okay, boleh faham Okay, so if I ask question, how many linkage group are present in a male? Untuk human, how many linkage groups present in a male?
24.
Okay, 24 linkage groups because 22 autosomal plus 1X, 1Y, so kita dapat 24 lah. Okay, so how many linkage group are present in a female?
23.
22.
20?
3.
23.
23.
Okay, so 23 because kita ada 22 autosomal. 1 because ada walaupun 2X tapi dia boleh dipanggil sebagai grup yang sama lah. 23. Okay. The genes are far apart on the same chromosome may independently assort from each other. Okay, this is due to crossing over. Okay, kalau ditanya soalan kenapa gene far apart on the same chromosome are independently assort from each other, so you have to elaborate about crossing over that happen during meiosis. Okay. Okay, if you find this word two-factor cross. Kalau jumpa perkataan two-factor cross. So it tell us about study linkage between two genes. Ada dua jenis gene, ada duk kaji. Macam mana dua gene ni bersama-sama ke, tak bersama-sama ke diwariskan. That one we call as two-factor cross study. Okay, if three-factor cross, so they are investigating three different type of gene, how they are closely or not closely inherited during the transmission. Okay, so dia ada banyak lagi 3 factor, 4 factor, 5 factor. The more the number, the more complex the study is. Okay, kalau banyak factor cross yang kita kaji, maksudnya study tu lebih kompleks lah. We need certain software to analyze. Kalau faktor tu banyak yang kita tengok. Okay. Okay, genes that are far apart. Ini yang dah ulang tadi kan. Okay, I have one example here. Two-factor cross. It is studies linkage between two genes. Yang kita akan tengok contoh selepas ni. It is a sweet pea plant. Okay. Ni yang Mendel cuba perhati. Okay, sweet pea plant investigating genes that encode for flower color, pollen shape. Sekarang kita fokus tengok dua benda. So that is two-factor cross. Okay, kalau three maksudnya kita tengok tiga karakteristik lah, tiga phenotype lah. Three phenotype that contributed by three different gene. Tiga different gene. As example, kita nak tengok the color of body of Drosophila melanogaster. Okay, the eye color and the wing size. Okay, so first is the color of eye. Second is the size of wing, and then third is the color of the body. Dia ada grey. This one case yang grey lah. Satu lagi dia mungkin boleh jadi warna kuning, yellow. Okay. And then eyes, ada yang commonly we can see red eyes. Okay. Satu lagi yang warna putih. And then in term of wing, bigger size and smaller size. Okay. Yang tu kita akan tengok secara lebih detail sekejap lagi. Sebab tu saya sebut terus apa ciri-ciri yang kita akan tengok. Okay. And then for sweet plant, okay, kita ada warna. Okay, saya terus jump kepada apa ni, yang pokok bunga. We try to investigate two genes. Two factor study linkage between two genes. Okay. We have this case, purple flower, and we denote as big P. Kita guna P besar. And then we have long pollen, big L. Okay. And then this one, red flower. Red flower kita guna huruf P kecil. Okay. Round pollen, small L. Okay. So capital letter or small letter, it tell us about something lah. Sama ada dia dominant ataupun recessive. Okay, kalau huruf besar kita untuk dominant, to show something is dominant. So in term of genotype for this one, it can be big P or big P small p. So it give this color. Okay, and then long pollen, it can be big L or big L small l. And how is about a genotype for red flower and round pollen? Okay, small p. Recessive gene only can show itself when it is combined with recessive allele. Recessive allele, jumpa recessive allele baru dia nampak lah dia punya phenotype, recessive phenotype. This one, Huruf apa kat sini kelas?
Small L,
L
small L.
kecil.
Small L,
kecil.
small L lah. Okay, good. Okay, big P, big L ni revision sebenarnya, revision. Sebab sebenarnya kita akan tengok benda yang lebih kompleks. So saya kena check dulu, kita kena buat revision dulu Okay, this one purple flower, long pollen. Okay, this one purple flower, long pollen. This one, macam mana kelas?
Purple
Purple
flower.
flower, round pollen.
Okay, good. This one?
Red flower, long pollen.
Red flower, long pollen. Okay. Ni?
Red flower, round pollen.
Okay, good. So, okay revision ni sebab dia ada kaitan genetic linkage, dia tengok balik yang you belajar Punnett square hari tu sebenarnya. Okay, this is a Punnett square calculator. Okay. It is okay to do analysis using Punnett square if the number of trait the number of trait is low. Contohnya 1 ke 2 trait ke 3 trait, it is okay lah. But when we are working with many traits kan, 3, 4, 5, the calculation will be more complex. Dia jadi lebih kompleks lah, and we need the software lah, kita perlukan software. Okay. Okay, just click here if you want to use Punnett square calculator. Just click and it bring you to this website, scienceprimer.com. Okay, this is example. Okay, you can move this slide. Okay, if you want to how many trait that you are looking at. Nak tengok berapa banyak kan, macam ni satu lah. Maksudnya ada A besar A kecil cross dengan A besar A kecil. What are the result? It will be automatically calculated for us. Okay, this one is easy. We can do manually on the paper. Okay, two also can, boleh jugalah. Nak tengok dua jenis trait lah sekarang ni kan. Kita tengok contohnya bunga tadi kan, satu warna kelopak, satu lagi bentuk dia punya pollen dia kan, dua trait. Okay, kalau saya tarik lagi jadi 3. 3 ni I think for me I have to start using calculator lah. 4, 5, okay, 5 it is more complex. Okay, jadi macam ni dia punya gambar dia and then it also can help us to know the frequency in term of ratio. Ratio dia in term of genotype, we can calculate in term of phenotype, so it help us a lot lah. Okay. Okay. So I hope you have learned about Punnett square. Okay. So as a continuity from what you have learned before, we would like to discuss about the finding by Batterson and Punnett. Ada dua scientist genetik ni, dia jumpa something yang unusual. Okay, so Batterson and Punnett, they conducted a cross in a sweet pea involving 2 different traits. Dia buat kajian tadilah yang saya tunjuk tadi kan, color bunga dengan bentuk pollen. Okay, so this is a dihybrid cross that is expected to yield 9:3:1 phenotypic ratio in the F2 generation. Okay, sepatutnya jadi when we try to do dihybrid cross in F2 generation, kita dapat phenotypic 9331 lah. That is from the original calculation. Okay, tak apa, sekejap lagi kita akan try kenapa keluar 9331 ni. Okay, after this we try to use the Punnett square. Okay, however, Batterson and Punnett obtained surprising result. They discovered 2 traits that did not assort independently. Means the result that they get is not like what they have expected. Dia tak jadi 9331, dia jadi ratio yang lain. Okay. Okay, we try to understand this picture first. Okay, tengok gambar yang kat atas dulu. Yang bawah ni tak payah tengok lagi. Tengok yang kat atas dulu. Okay, first, the parent generation. Okay, we have big P, big L, big L. Okay, means they have the phenotype like this. We try to hybrid with small p, small l, okay. So what happened to F1 generation? Okay, all flower will be like this in term of phenotype. Dia akan ikut generasi satu, dia akan jadi rupanya macam yang ini. Why? Because big P only will combine with small p. Big P will not be able to combine with another big P. Sebab kawan dia ni, bini dia ni kan dua-duanya kecil-kecil kan. Ni pun L pun kecil-kecil. So we will get F1 generation like this. P besar, P kecil, L besar, L kecil. So all F1 generation will be purple flower and long pollen in term of phenotype. And in term of genotype, all of the flower will be big P, big L, small l. Okay, boleh setakat ni? Boleh
Boleh.
Kenapa
Boleh.
semuanya warna purple? Kenapa semuanya long pollen? Okay. And then we try to do hybridization. We try to hybridize members in F1 with another member also in F1 lah. Maksudnya F1 anak dia ni kacuk dengan anak dia juga dalam ni kategori ni. Okay, so we try to do this. We try to do big P, small p, big L, small l. We try to hybrid with big P, small p, big L, small l. Kita cuba buat. Kita nak tengok apa ratio yang akan jadilah. Okay, so saya tunjukkan. Saya cuba tunjukkan guna ni lah. Kan? Punnett square ni. Okay, so kita pilih berapa trait kelas tadi? Nak guna contoh tadi? Berapa trait nak pilih? Nak buat yang bunga tadi?
2.
Okay, 2. Okay, tapi ni contoh A dengan B. A dengan B saya tak nak. Saya nak this one lah kan. Okay, parent 1 parent 1. Okay, I make it bigger. Saya cuba buat besarkan sikit. Okay. Is it bigger now? Or same?
Yes.
Yes.
Okay, good. So we have parent 1, we have parent 2. Parent 1, what is the trait? Apa saya nak key ni? Big P and saya nak kacuk yang ni. Saya buka balik. Saya nak kacuk ada ni cerita dia kat sini. Saya nak kacuk yang ni F1 dengan F1 yang lain. Maksudnya nak tulis apa? Kat sini nak tulis apa?
Kecil.
P kecil lah kan? This one?
L besar and L kecil.
L besar, L kecil.
Okay, this one. Parent number 2.
P besar, P kecil.
Okay, good. Maksudnya you faham lah apa yang kita buat. Saya kena tanya sebab kalau saya nak tahu student tanya ke tak, faham ke tak, saya bertanya je lah. Okay, this one?
L kecil.
L kecil sebab kita cuba kacuk F1 dengan member F1 juga kan. So we try to show the frequency. So we are entering 2 trait cross. Okay, we have 16 square. Dia akan kira lah, dia akan beritahu lah 9 unit allele combination. Okay, and then it count the genotype, the count, the percent, okay, and then we can see that mana ratio dia? Sembilan tiga satu. Oh, dia tak mention Sekejap 9331. Okay, this one we have to mention about the dominant allele we have to inform the computer, the calculator. The dominant allele is big P, dominant allele is big L. Okay, so it tell us the ratio of the phenotype is 9:3:1. Kita akan dapat 9:3:1 lah. Tu yang what Mendel and Bateson expect. Tu yang dia expect 9331 lah bila kita buat ni. Tapi bila tengok result, okay, they surprise the result that we get is surprising result. Okay, so now we look at this expected number and ratio. Okay, this is the number lah, berapa banyak yang karakter macam ni dia kira, and then from this number it can be transferred into this format lah, dalam bentuk ratio ni yang kita expect. But when we look at the actual data, the ratio it is like this. Dia jauh lagi. Okay, this one it is more common. This one is more common. Ni yang lagi naiklah nilai dia kan, daripada 1 jadi 45, daripada 9 jadi 15.6. And this one it become lower. It tell us that what we get from here, this one and this one it is more common. It is like they are inherited in unit. The rate is more common. Okay, dia lebih banyak kali berlaku macam tu. Okay, so what Bateson and Pandit find, it show that independent assortment does not always occur bila dia tengok pada apa ni trait yang this one, dia tak sama macam kajian dia trait yang lain. As example, they are try also look at the trait, the height of the plant, the color of the pollen. Tapi dia tengok dua karakteristik ni dia tak ikut. Okay, dia tak it showing that independent assortment does not always occur. Dia tidak berlaku secara rawat, dia seolah-olah macam dia memilih-milih. Macam tu. Okay, so two genes it control flower color and pollen shape are found on the same chromosome. Okay, so the offspring it tend to inherit the non-recombinant pattern. It tend to inherit non-recombinant pattern, whether big P, big L, or small p, small l. Dia macam lebih nak ikut dia punya ni, dia punya atuk dia ni, atuk dengan nenek dia yang asal. Okay, sebab kita panggil sebagai apa ni, parent combination lah. Okay, dia nak ikut macam keturunan dia yang asal, dia punya pattern dia, walaupun kita tengok dekat F2. Okay, F2 dia macam seolah-olah dia nak juga ikut macam ni kebanyakannya lah. Macam tu. Okay, due to occasional crossing over, a lower percentage of offspring inherit a recombinant pattern of allele. Ada juga dekat ni, this one kan. Ni. Berlaku juga, bukan tak berlaku langsung. Yang selain daripada ikut nenek dengan atuk ni. Okay, ada juga yang ikut this one jadi big P small L, small p big L, but in term of percentage it is less common. Okay, boleh faham ke setakat ni? Kan ada soalan?
Faham?
Okay, good.
Okay.
I have to open. Oh, now it is around 1 hour. Dah sejam dah. I think that I have to stop recording and I start again the recording lah, because I don't want to make the recording very long. Sebab kadang-kadang kalau you all nak download balik ambil masa yang panjang kan. So I try to start the recording first. I want to separate into 2 files lah. Okay, kat mana?